Muscle Tutorial by Vincent Akridge and Sam DeVito. There are three types of muscle: skeletal, smooth, and cardiac. Skeletal muscle's location is self-explanatory. It is found attached to bones by tendons. Smooth muscles are found within the walls of hollow organs, and cardiac muscles are found within the heart. The following series of images were dissected from these specific areas of our rat.
Each muscle type has certain characteristics that can be differentiated from one another. For example, skeletal muscle has clear striations with the generally separated nuclei along with its muscle fibers. Cardiac muscle is also striated, however, it has intercalated discs and the branching striations are not the same as the muscle fiber striations. The nuclei are surrounded by glycogen. And lastly, smooth muscle is not striated and has spindle-shaped cells.
All muscle movements are controlled by the brain via a nerve impulse. Here's how it works. When a nerve cell needs to send a signal, it opens special channels in its membrane. Sodium ions rush into the cell, making the inside more positive. This creates a wave of electrical activity that travels down the nerve cell. After the spark passes, potassium ions flow out of the cell to restore the original charge balance, thus causing a muscle to react. Now, there are two types of muscle reactions: voluntary and involuntary. Voluntary reactions are those that you consciously control, such as lifting your arm or wiggling your fingers. Involuntary reactions are those that you don't control, but they still happen. We tested involuntary reactions in an experiment. We each took turns throwing a ball at each other and counting how many times we blinked. Here's a sample of the experiment. Once the ball hit my face, my body forced itself to blink because it sensed potential danger. Even though I didn't consciously control that reaction, the same process still occurred.
Muscular contraction requires several key components. However, we will be focusing on ATP, adenosine triphosphate, and salt for this section. For muscular contraction to occur, both ATP and salt must be used. The body can't simply use one of them because the muscle won't contract without the other. Let me give you an example. This is how muscle fibers react to just salt. This is muscle fibers with just ATP. Now the muscle fiber with both. Muscles need both salt and ATP because salt alone cannot trigger the necessary electrical signals in nerve cells within the muscle fibers, which is why it requires the assistance of ATP to contract the muscle.
Muscle tissue consists of many structures, so we'll break them down individually. Starting off with tendons, these are tough bands of fibrous connective tissue that connect muscles to bones. They help transmit the force of muscle contraction to the bones, allowing for movement. Next is the epimysium. This is a layer of connective tissue that surrounds the entire muscle. Its primary purpose is to hold the muscle fibers together. The deep fascia is another layer of connective tissue that surrounds and supports muscles, blood vessels, nerves, and other structures. The perimysium is yet another layer of connective tissue that holds bundles of muscle fibers called fascicles together and provides support and protection. The final layer of connective tissue is the endomysium. These surround individual muscle fibers, thus also providing support and protection. Muscle fibers are single muscle cells. They are long, cylindrical cells that contain many myofibrils. The sarcolemma is the cell membrane of a muscle fiber. It helps regulate the flow of ions in and out of the muscle fiber. Myofibrils are long cylindrical structures that run the length of the muscle fiber. They are made up of myosin and actin. Finally, a motor neuron is a nerve cell that stimulates muscle contraction. When a motor neuron is activated, it sends an electrical signal to the muscle fiber, which causes the muscle fiber to contract.
The processes of muscular contraction is relatively extensive, which is why we will walk you through this in a series of steps. Step 1. As a nerve impulse reaches the end of an axon terminal, it stimulates the release of acetylcholine from synapse vesicles. The acetylcholine will then diffuse across synaptic cleft and attaches to acetylcholine receptors on the sarcolemma. at the neuromuscular junction. Step 2. After acetylcholine attaches to the receptors on the sarcolemma, the muscle fibers become more positively charged. This generates an action potential that travels along the sarcolemma and into the transverse tubules. Step 3. As the action potential travels along the T-tubules, calcium is released from the sarcoplasmic reticulum.
Step 4. Calcium binds to troponin on the actin filaments of muscle fibers. This signals tropomyosin to move out of the myosin binding sites on actin. Step 5. Energized myosin heads with an attached ADP molecule and phosphate group bind to the open myosin binding sites on actin. This forms the actin-myosin crossbridge. After the crossbridge forms, the myosin head rotates towards the center of the sarcomere. As it rotates, the myosin releases its ADP and phosphate. Finally, step 6. After the power stroke, the actin-myosin crossbridge remains intact until the ATP molecule binds to the myosin head. After ATP attaches to myosin, the myosin detaches from actin.